you, dear students. Me is Dr. Basanti Shukla from Department of Zoology, Kendrapada Autonomous College. Okay. And I think, should we start or wait for some times? At 9:05 a.m. we will start our class. And our syllabus is from 137, Block 1. You might have got your booklets, students, dear
No,
students.
ma'am.
No, ma'am.
No, ma'am.
No,
Oh,
ma'am.
so from which you are from? Plus three first year, second year, third year?
Ma'am, second year.
Plus three second year. Till yet you have not received your booklets.
No, ma'am.
And may I know your residential city?
I am from Dhenkanal.
Your name
Ma'am,
is
Brahmpur.
Barampur, Dhenkanal. And may I know your academic or professional careers? Tell me
Ma'am,
one
I
by
am a
one.
student.
Hmm.
Ma'am, I just completed
Which,
my DLED
mhm.
and continuing my
Yourself
plus
is
3
Rupali.
in regional.
Rupali.
Ma'am,
Name. Tell your name, please. Rupali
Anuradha
Priyadarshan.
Padhi. No, ma'am,
Tell me
Anuradha
now.
Padhi.
Anuradha Padi. Anuradha, you are from which
Brahmpur,
place?
ma'am.
Oh, Barampur. Particular Barampur. Near that university
No,
campus.
ma'am.
Hmm. Acha, you have just completed your
DLED, ma'am. Secretary.
Aniram. Okay. And another one is Kabita Pradhan.
Yes, ma'am. Good morning,
Kabita?
ma'am.
Yes,
Yes,
good
ma'am.
morning. Very good morning. Where from Kabita? Which place?
Ma'am.
You are from which place?
Jharsuguda district, Banbahal.
Okay. And you have completed your what type of academic careers?
Ma'am, actually, I used to after marriage, I used to live in Bangalore, where I was continuing B Farm. So,
Very.
because of some, mat- my husband got shifted here to back to Odisha. He got job in MCL, so we
Mm.
came back, and I
You
decided to
join.
continue from distance course.
Oh, that means you have completed. Mm. Mhm. And dear students, you should write your roll number in our chat box. Okay. And what's about Sachin? Sachin, you are the first. Okay. Hello.
Hello.
Yes, Sachin. What type of career
Yes,
you
ma'am.
have completed?
Ma'am, I have completed my DLED city
Mm-hmm.
after plus two. Then I enrolled to BSCG in IGNO.
Oh, no. Okay. Then Devasmita, please write your roll numbers. I have to take those your roll numbers present. Today's present. Okay. Attendance. So write your roll numbers, please. Students, write your roll numbers in chat box. So I'm going to deliver my lecture
Yes,
on
ma'am.
that. Yes, okay. Right. And I'm going to deliver the lecture on that 137 block 1. That's the genetics. Okay. And this genetics means what is its meaning? Genetics, it's the study of behavior of genes and transmission of genes. Okay, students. So after the introduction of this genetics class, I would share my screen with you and let me know the whether it is visible to you or not. Okay. Just I am
Yes, ma'am.
sharing. Okay, thank you. I am sharing my screens to you. Write it to the chat box whether it is visible to you or not. Is it visible?
Yes, ma'am.
Yes,
Monohybrid
ma'am.
cross.
Yes, thank you. So before going to that monohybrid, dihybrid, and trihybrid process, and I hope you have completed your these um, academics and text during your plus two and also in high school level also. This genetics that refers to the study of genes, its behavior and its transmission to the generation after generation. So, the father of genetics, who is father of genetics? Okay,
Gregor
Gregor.
Mendel.
Yes, John Mendel, Gregor John Mendel. He has done a lot of experiments in pea plant. Okay, and as this pea plant has a numerous effect, or we have that opportunity to produce a lot of generation within a short period. That's why he has accepted that selected this pea plant um, for hybridization or the selection of experiments in genetics. And what happens in genetics actually during period of Mendel before the discovery of DNA, and he used it as what? Not the gene, the factor. There actually the some scientists they use as factors, and they have observed that some factors that inherited from generation to generation, and after discovery of the DNA, that factor was named as gene, and this gene is the unit of character or unit of evolution. And this gene is a chemical composition of a small fraction of a DNA segment. And students, you know DNA. What is DNA? This DNA is present in chromosomes, and chromosomes are the hereditary materials present in nucleus. Is it or not? The chromosomes. So they present in nucleus, and the DNA is known as the genetic material of the nucleic acids. Okay, and this DNA or the fraction of the DNA is the gene that how that inherited from generation to generation. So these genes are the alleles. Alleles, what type of word is known as alleles? Alleles are the similar or the a modified form of the genes present in homologous chromosomes. And you might have known the homologous chromosomes means a pairing of chromosomes or a set of chromosomes. They are inherited from both parents, and they are similar in origin and structural behavior. Okay, and in homologous chromosomes, the genes present in a particular locus or loci. Singular form is the locus and plural form is the loci. When in a particular position of two chromosomes, genes present, so these are known as the alleles. For example, the for tallness of the pea plant, the capital T is used. That is, capital T is also one allele from one pair, and small t that represents the dwarfness of the gene, or that represents the dwarf gene. So in this screen, just see heterozygous mother female gamete, and R represents the tallness or the dominant capital R, that is for the dominant capital R is the dominant for round seed, and small r represents the wrinkle cell. Okay, that round seed and wrinkle seed, if two mothers are mated or crossed, one parent, one male and a female with a round seed and a wrinkled seed. Here, wrinkled seeds are the recessive character, and round seeds are these are dominant. And then what happens in the gamete formation? The all the in first filial generation, all the seeds will be round. Okay, because round is dominant character, and in Mendelian laws, the law of dominance represents that when two allelic genes are inherited, during that time the dominant gene that conceals the effect of the wrinkle character, but it is not always. In the first filial generation, that expresses itself or marks the dominant character, that marks the recessive character. Okay, so in first filial generation we are getting, okay, in what happens in second filial generation, that dominant character and a recessive character, they express themselves, but a mixed variety, that is in the dominant form but heterozygous form, that is expressed in first filial generation. And in dihybrid cross, what happens when two contrasting characters? In monohybrid cross, we have taken a single contrasting character, that is wrinkle and rounded shape. And in case of the dihybrid crosses, two characters, two contrasting characters, means the fur color. If we take the example of guinea pigs, so fur color black and white. Black is always dominant over white fur color. And in coat texture, that is another character, that is the texture, means the texture of the hair. Are you comfortable, students? Are you comfortable in
Yes.
English? Okay.
Yes,
Yes,
ma'am.
ma'am.
Okay. Any other student from outside Odisha? Is there any other from outside Odisha?
Yes, ma'am. I am
Okay,
outside,
thank
no?
you.
I'm
So
Sambalpur.
here in, oh, from Sambalpur. Okay. Are you comfortable in English?
Yes, yes, yes. Okay, no problem.
Okay, thank you. Everyone is comfortable?
Yes.
Or should I use sometimes in Odia version? Okay, okay. So should I continue in English? So coat's texture means the texture of the hairs. That is a rough one, and another one is smooth one. And rough one is always dominant over smooth one. So in first filial generation, four types of gametes are formed. That is capital B black with rough one, that is a pure dominant variety gametes. that will be formed, and another gamete form is that is black with smooth, black with BR form, and the third one is the recessive smooth with um, rough feather or coat texture with the white color or recessive color, and fourth one is the totally recessive, both recessive gametes, that is small b and small y. So let us cross among themselves. So when we are getting dihybrid crosses, we are getting 16 varieties, and just you see here 16 varieties of offsprings are there, and among them just see one is the pure variety, that is the first one, capital B, small no, capital R. Okay, that is the pure variety, and another one is the pure recessive one, that is the small b, smaller, and here in this form 16 varieties are formed. Phenotypes are 16 varieties, but genotypes are 4 varieties. Just you see here, observe here, the genotypes mean genetic configurations are 4 types, but varieties, phenotypes are 4 varieties, but genotypes are 16 varieties we are getting here. Okay, come to the next slide. In after dihybrid, so dihybrid plus, in here 3 contrasting characters are taken, and here 64 varieties in Punnett square tables. We are getting 64 varieties having 3 uh, contrasting characters. Okay, uh, and in this dihybrid cross, see here 3 type 6 how 64 varieties are here having the 8 varieties of gametes. Okay, when we are crossing them, we are getting 64 varieties having 3 contrasting characters. This is known as the trihybrid crosses. And what happens in law of segregation, actually segregation means to segregate, to be separated. Okay, so what happens in the yellow or green, or that coat color, maybe fur color, or the roundness or wrinkleness of the seeds, so everything they are segregated from each other. Okay, and each gamete carries just one of each gene pair. When the offsprings are conceived, each receives one gene from each parent. That means they are separately and getting the separated forms of the genes from each parent. Okay, so in laws of segregation, what happens? See here in the screen, the small y, and when we are crossing the small y with the capital Y, the dominating nature and the recessive nature, we are getting one gamete, that is the mixed variety, and this dominating character that marks the mass, or that conceals the expression of the recessive variety. But that doesn't mean that always that recessive variety, because we have observed that in dihybrid cross, monohybrid cross, and trihybrid cross also, that the recessive character is also expressed separately when it is being allelic pair or jointly they are expressive in a gamete. The dominant character that conceals the character, but that doesn't mean that it is destroyed. So recessive character is also that is expressed in the further generations. That means the law of segregation. Okay, and in this experiment, a test cross is there. When test cross means when you use to determine a phenotype of an unknown dominant individual. Uses of homozygous recessive individuals are the test. Means the first F1 generation offsprings, if they are mated with the parental the parental combination, or they mate with the parents. So this is known as the back cross. But when the F1 generation offsprings, they mate with the purely recessive parents, that is known as the test cross, homozygous recessive. When we are mating those homozygous recessive parents with the F1 generation offspring, so we are getting the that example of the test cross. Okay, the law of independent assertion means independently how they are asserted. Each individual allelic forms or the genes that have the power to be expressed independently. That means when what happens when we cross a brown-eyed person with a blue-eyed person, and blue eye is always that is dominant in nature. So what happens, see here, states that genes for different traits, for example eye shape and eye color. So here these are independently the inherited genes, or genes they inherit themselves independently from generation to generation. Okay, brown eye and the um, round eye. So the first gamete formation will be, that is the round and brown and round color. Okay, so here, so independent assortment, one is homozygous trait and another is the heterozygous trait. So in homozygous, what happened? The two genes they are present, and heterozygous means first filial generation when they are produced, one dominant with another recessive genes. These are known as the heterozygous. So different genes are present. So homozygous and heterozygous examples are there. Okay, here phenotype, if that is present represented phenotype in blue eyes and the brown eyes. Here in case of the blue eyes, genotype is small b and small b. Blue, because blue eye is recessive in character, so this is genotype, but phenotype it looks like a blue eyes. In brown eyes, one is one gene, that is small b, is inherited from the blue eyes, but genotype looks small b capital B or capital B capital B, but phenotype, and in that phenotype means physical appearance, that looks brown, but it may carries that capital B small b configuration or capital B configuration. Okay, then come to the next. And in it two what of that is the complete dominance and incomplete dominance. When dominant allele completely masks, I have told you that masking of the dominating character over the recessive character. So in complete dominance, the dominant allele completely masks the recessive character, that is known as the complete dominance. But what happens in incomplete dominance? Neither allele is dominant. And here what happens in red color, total capital R's are and small r's, they are neither dominant. So here the configuration is capital R and small r. In this case, They may be a mixed variety or pink variety. That is also example in mirabilis plant, that is the 4 o'clock plant. Okay, so that is the pink variety. Neither red is expressed nor the white is expressed. So in codominance, one allele for a trait is not dominant over the other. So they both will be expressed in codominance. So in here, what happens when the red that is crossed with the white color in incomplete dominance, that comes as pink color, but in codominance the mixed uh, red color and also the white color, they are expressed in flower petal colors. That is the example of the mirabilis plant or 4 o'clock plant. You might have seen that flower. Okay, come to the next. As here is also that example, codominance. Both will be expressed. The blue color here, blue color and albinism white color. So both are expressed. No one is concealed by the other. That is known as the example of the codominance. And in multiple alleles forms in blood type. And the blood grouping forms are A, B, AB and O types. Okay. In blood typing 8A allelics, the genes they are present in genotypes. In A blood type, genotype is IA and IB, both are codominant. Okay. The third allele is recessive. The blood typing genotype in O group, that is the IIL's O-type group when both alleles are II and AB when AB both are expressed, that will be the blood grouping AB. And in case of the both IA array, and that will produce the blood grouping A type. And in case of the B type, we are getting forms B or BI forms. Okay, so these are the alleles present. And in case of the RH inheritance, that is independent of the varieties A, B and O type. In RH factor positive, uh, in case of a person, so RH factor possible genotypes may be both positive or RH1 or RH negative, and RH positive is dominate over the RH negative. Okay, and in case of the in next table, parent 1 and parent 2 and the types of child. When parent 1 has RH positive, parent 2 has RH positive, so child must be RH positive. When one parent is RH negative, another parent is RH positive. So RH positive is as it is dominant over the RH negative, so child will be RH positive. When both are RH negative, the child will be RH negative form. And the gene interaction that produce new phenotypes, what happens? This interaction in maintains dihybrid process. What we have observed, the genes for different traits acted independently for independent assignment, because we have discussed that law of independent assignment. The genes for different traits acted independently. That is, the allelic pair for smooth or wrinkled cells had no effect on the allelic pair for long or short stem. So these two varieties, they expressed independently. Okay, and however, there are cases where two allelic pairs affect the same phenotypic characteristics and non-allelic genes interact. Am I audible, students?
Yes,
Hello?
ma'am.
Okay,
Yes,
thank
ma'am.
you.
Yes.
Okay, thank you. When two allelic pairs, we have already discussed that the same loci In a homologous pair, the same loci genes, they are known as the allelic pairs. And when should we call that non-allelic? Means different places. The genes, they are present different places, not in a parallel position. You might have, you should refer your Google searches also. What is the allelic pair and non-allelic pairs? And for that, I have not made that table. So, okay, in a parallel form or a similar loci or position, locus, location from location, that is we are using the loci. So, however, there are cases where two allelic pairs affect the same phenotypic characteristics and non-allelic genes interact to give new phenotypes and result in a modified phenotypic ratio. Okay, and a classic example of such gene interaction is comb shape in a chickens. उन लोग का हेड में जो कॉम्ब होता है ना कॉक का, okay, that comb shape in chickens. So there are 4 comb shape phenotypes which result from combination of alleles are 2 different loci, different locations or different loci. So one is rose comb results from the combinations just you see here. Okay, so one is rose comb, another one is walnut, pea comb, walnut, and a single comb. Okay, these are the phenotype. See here what type of 4 types of comb morphologies here. I have made for you this. Okay. Next, come to the next. What is epistasis? Epistasis means one gene completely masks the another gene. Okay, so dominant gene when both allelic genes they express in one generation when they present side by side. So the one that masks or conceals the another one, that is the process is known as the epistasis. Okay, and what type of lethal alleles are there? Some examples are there. Lethal, these are dangerous for the generation or the organism. So allele which cause an organism to die is known as the lethal allele. So certain genes are absolutely essential for survival. In mutation in these genes create lethal alleles because mutation occurs during the crossing over. Okay, later we will go to that topic. So what happens when mutation occurs? Some lethal alleles are being created. So lethal alleles are maybe dominant or recessive. So only dominant lethal allele kills the organism in both homozygous and heterozygous condition also. Certain lethal alleles kill organisms in homozygous condition only. Types of lethal alleles. These are 4 types, 4 categories are there lethal alleles: early onset, late onset, conditional, and semi-lethal. Early onset allele means lethal alleles which result in death of an organism at early stage of life. For example, during embryogenesis, the very early stage. Okay, so during the embryonic stage, that allele is produced. In late onset, lethal alleles that onset due after onset or final stage of life, that is known as the late onset, when that allele kills the organism, okay, in late hour of the life cycle. And conditional means lethal allele which kill organism under certain environmental condition. Those are the examples. What happens? That is the temperature sensitive alleles kills organism at high temperature or during the sunstroke time also. That is one type of allele. So when body is not able to tolerate the environmental condition. And in semi-lethal condition or alleles, semi-lethal alleles, which is kill only some individuals of population, but not all the individuals. In a population, some individuals are killed by this lethal genes. And another one in your subject, that is pleiotropy. Pleiotropy means a single gene can have multiple effects on phenotype. A gene can express itself for different diseases or phenotypic disorders. So, if we take the example of a normal person with a sickle cell anemic person. A normal person can be mutated. Okay, so signs and symptoms means one allelic or one single gene that may create or that may be responsible for different phenotypic symptoms or phenotype. That means, example, multiple symptoms of sickle cell anemia. That means that is giving the pain, or that may create jaundice, that may create different infections or fatigue or tiredness. Okay. Um, so sex-linked genes, and you all know that sex-linked means in case of the humans, that is the X and Y chromosomes are known as the sex chromosomes, and Y chromosome is limited for some expression of the Y chromosome linked diseases, but X is has a wide variety of linked genes for the expression in several diseases or disorders. And in this condition, that the penetrance and expressivity of some diseases are the sex chromosomes. So, in what is penetrance and expressivity? Penetrance means percentage of individuals with a given genotype who exhibit the associated phenotype with that genotype. But here expressivity means the intensity of the phenotype in an individual. Here penetrance, that is the individual with a given genotype, the particular given genotype that exhibited, and a measurement, how we will measure that in a, that is in a population. Penetrance example in a population, but expressivity that in a single individual. Variability, as per the variability, statistical variability among a population of genotype, and it is in a, so we are comparing, that is penetrance in a population, expressivity in a particular individual. And come to the next one. Just a minute. Okay, then come to the unit 3, that is the determination, sex determination. Sex determination means how to determine the sex, or how the sex is determined from the embryonic stage and expressed in the formation of the individual. That means the mechanism of sex determination or genotype of sex determination system, in which sex is actually governed by genotype configuration of the zygote, and its environmental sex determination also, that present in which sex is governed by internal or external environmental condition. So sex determination may be genotypic, or that may be environmental condition or influenced by the environment. Okay? In humans, what happens, and other placental animals, the mode of sex determination is the Y chromosome mechanism. In case of the human, that is XY, that denotes or that helps in the formation of a boy child, and in case of the double X, that is a female child is born. Individuals carrying the Y chromosome are genetically male, and they are lacking when double X are there, so they are called as females. Okay, and the product is called the testis determining factor. That is the testis determining factor causes the gonadal primordia to differentiate into testes instead of the ovaries. And in case of the male and female Drosophila, what happens? That is also X chromosome and Y chromosome mechanism. In birds, that is determined by JW female. It is female is heterozygous, but in case of the humans, we are getting Drosophila and humans. Double X means female. Here that means in case of the Drosophila and of humans, they are homozygous, but in birds, heterozygous condition is denoted as the females, and homozygous that denotes for the male product. So females have one Z and one W chromosome, produce two types of eggs here in case of the birds. So the males are known as the homomorphic Z chromosomes in case of the bird, and XX female, X0 male, that is found in case of the grasshoppers. Females are both X chromosomes are there homogametic, that means they produce similar gametes. Okay, X and X gametes. But here one is only X chromosome is present in case of the grasshopper. Okay, so zero or XO chromosomes, that is known as X0 or XO chromosomes. So absence of one chromosome. Okay, only X chromosome is present. So another partner is absent. So in grasshoppers and proteiners and many other insect forms, we are taking that XX and X0 example of sex determination process. And what is ploidy? Ploidy means the number of bases of chromosome sets. In a diploid set means diploid organism has two sets of chromosome. Example gives the 23rd into 2. We have two sets. So we are diploid animals having two sets of chromosomes, and a diploid has two sets, whereas a hexaploid has 6 sets, and a monoploid has one set chromosome. So the basic chromosome number X, also called the monoploid number, is the number of different chromosomes that make up a single complete set. Ploidy can be divided into two as euploidy and aneuploidy. Okay. And what is euploidy and aneuploidy? Euploidy means the condition of an organism having complete set of chromosomes or multiples of the basic set is euploidy, and the cell is called as the euploid cell. When aneuploid means the condition of chromosome number is not an exact multiple of the normal diploid number, with either fewer or more than the normal number. Here it is divided into euploidy and aneuploidy. Ploidy is divided into euploidy and aneuploidy. Euploidy is again divided into monoploidy and polyploidy. Here this polyploidy is autoploidy and alloploidy. It is okay, and allopolyploidy and autopolyploidy. And aneuploidy means hypopolyploidy, hypoploidy and hyperploidy. Hypo means that is the lesser, and hyper means the higher structure or higher expression. So in hypopolyploidy, monosomy and aneuploidy, and here hyperpolyploidy, that is trisomy and tetrasomy forms. Monopolyploidy, an individual that contains one half of the normal number of chromosomes. That is monoploid or exhibit monopolyploidy. And that exhibits the monopolyploidy. Some species such as bees, ants, and male bees are normally monopolyploid because they develop from unfertilized eggs. When they are developed from unfertilized eggs, so they express that monopolyploidism. Okay. Consequently, the individuals are sterile. Monoploids are very rare in nature because recessive lethal mutations become unmatched and thus they die before they are detected in case of some monoploids. These alleles normally are not a problem in diploids because their effects are masked by dominant alleles in the genomes. So these alleles actually normally these are not detected, and in polyploidy what happens here, they may be they are more than two homologous sets of chromosomes. Very common, these are found very common in plants associated with origin of new species and or may positively correlate to size of individuals. So there are two classes of polyploids based on the chromosome of chromosomes, even number, and another one is odd number. And what is autopolyploidy? That results when all sets of chromosomes are from the same species. This is known as the autopolyploid. Examples for autopolyploids like the grapes, bananas, and some garden plants or allopolyploidy. These are closely related species. So the hybrids, they are from very closely related species. And another is homologous or partially homologous chromosomes. Allopolyploidy is caused by the presence of several genomes of different species in a cell or tissue or a whole organism. Examples: fusion of haploid gametes from species 1 and species 2 produces N1 plus N2 hybrid plant. No chromosome pairing occurs here at meiosis, and the viable gametes are not produced and the plants are here sterile. And in case of the aneuploidy, this is a condition where the chromosome number is not an exact multiple of the normal diploid number, with either fewer or more than normal number of chromosomes in the cell. And these aneuploidy these are the causes. A condition of aneuploidy may occur due to loss of one or more chromosomes during mitosis or meiosis. So due to loss of one or more chromosomes and due to non-disjunction or irregular, that is the irregular distribution of sister chromatids. Here in case aneuploidy, just you see hypopolyploidy and hyperpolyploidy. In hypopolyploidy, monosomy and nolisomy, we have already discussed that in that ray diagram. So in case of the hyperpolyploidy, trisomy and tetrasomy occurs. And in monosomy, it is a form of aneuploidy with the presence of only one set. Already we have discussed it, monosomy. Okay, only having the one set of chromosomal pair, and the monosomic cell hence involved a loss of single chromosome that is known as that may be 2n minus 1, and partial monosomy occurs when only a portion of the chromosome, a part of chromosome has one copy while the rest has two copies. In case of the nolisomy, that involves loss of one homologous chromosome pair, that is the 2n minus 2, it is also lethal in deep blood. It's very lethal and can be tolerated in bred wheat. It's a hexaploid, behaves meiotically like a diploid. 4 homologous chromosomes compensate for missing pair of homologous. Okay, and in case of the trisomy, a chromosomal anomaly that is characterized by presence of extra chromosome in the cell. Presence of extra chromosome. So here it is found as trisomy forms. That may be an example of a person, or that those are found in also plant cases. And in trisomic cell involve extra chromosome that is present in 3 copies of one chromosome type, and that is the example as 2n plus 1. So in case of the trisomic individuals in human cases, Down syndrome, and trisomy that is in not that is present in autosomal pairs. Pair number 21, trisomy 21, so that expresses Down syndrome, and in the karyotype of 18 pair trisomy that gives the Edwards syndrome, and the trisomy number 13 that is Patty's syndrome. And in case of the tetrasomy, the condition tetrasomy cell involve extra chromosome pair resulting in the presence of 4 copies of one chromosome and 2 copies of every other chromosome type. So tetrasomic cell genotype will be 2n plus 2. Okay, just a minute. I'm giving another slide. Yes, in sex linked, I discussed that the sex determination, trisomy, monosomy, and euploidy, aneuploidy. So here, some examples are sex linked inner. Some genes are there linked to some particular sex. Okay, so in case of the chromosomes that are the carrier of the genes, so some autosomes and sex chromosomes, because we know so in the karyotype study of human beings, 22 pairs of autosomes are there, one set of sex chromosomes are there. So in certain genes present in the sex chromosomes controlling the somatic character. The character which are controlled by sex-linked genes. So the characters will be sex-linked characters, and these are their transmission from generation to generation is known as sex-linked transmission characters. So this is known as the sex-linked inheritance. And in sex-linked inheritance or sex-linked characters, so some doses compensation term. Doses compensation means this refers to a regulatory mechanism that ensures the equalization of sex X-linked gene products in males and females, because X is present, X chromosome is present in female and in male also. Okay, so that in doses compensation happens in case hypertranscription in Drosophila. Just see here, double X 1 plus 1, and here the X is known as the doses compensation double dose here, because that gene is present in both the chromosomes. And in X inactivation mammals, when in case of the male, the Y is present only single X. So here a single dose is present in case of the mammal males. And hypotranscription in case of the senior hybrids, that is the X and X present in half configuration. Okay, so here some X-linked recessive traits in humans. X-linked, but their character is recessive in nature. That is, recessive characters are color blindness, hemophilia, and the muscular dystrophy, and SCRD means severe combined immune deficiency syndrome. So these are some X-linked but recessive in traits. And X-linked dominant traits present in case of the humans. These are the affected males produce all affected daughters and no affected sons. Okay, so because X-linked dominant means X would be dominant in nature, so naturally they would transmit that character to the daughters. Okay, and no son is affected by those fathers. A heterozygous affected female will transmit the gene heterozygous female, and that is affected by that gene transmit the gene to the half of her children, male or either male and either female children. And about twice as many females as males are affected. Females and males are affected by those X-linked dominant traits. Few of these traits are also known in case of the humans. Some Y-linked traits are Y-linked traits, hence those will be affected or transmitted to the sons only. So in this case, most Y-linked mutations lead to sterility and cannot be inherited to all the. So here it is known as the hypertrichosis. See here how the hairs are more developed, facial hairs and ear pinna hairs. So it is one example of the Y-linked trait. And the sex limited inheritance. Sex limited inheritance, that is in case of X or Y chromosome. That is known as the example is hemophilia, and sex limited that comes under the milk production in cattle, and sex influenced genotype and sexual determinants, that is the horns in most sheep and beards in some goats. The chromosomal theory of inheritance. So, in chromosomal theory, what happens? Chromosomes and genes are present as pairs in diploid cells. Homologous chromosomes separate during the meiosis, and this separation of homologous chromosomes again they restore their chromosomal or diploid nature or recombination or restoration during the fertilization, because two haploid gametes they mix and again restore the diploid nature of the animal. The chromosomal theory of inheritance claims that it is the chromosome that segregates and asserts again independently and repeating the important laws of the Mendelian hypothesis. So here chromosomal theory in inheritance, certain and very propose the chromosomal theory of inheritance. According to this theory, it is well established that many genes are located in each chromosome in a linear fashion. In a linear fashion means in a linear fashion, in a similar loci of the homologous pair chromosomes. And all genes located in some chromosome would move to same pole during the cell division. That is during the what? Anaphase and telophase. So they are moving, all genes they are located, so they move to the same polar locations, and as a consequence, such genes will fail to show independent segregation and will tend to be inherited together. And students, now time is over, so we should stop here. Thank you, students.
Thank you, ma'am.
ma'am.
Thank you. Okay,
Thank
thank
you,
you.
ma'am.
Okay, thank you.
Thank you, ma'am.
Students, those joined lately, please write your roll numbers in chat box. Again, you write.
Yes, ma'am.
Yes, please write your roll numbers in chat box. PK Sahu, write your roll number, please. Hello. Yes, you have already. Numbers, please. Those joined lately, give your roll numbers. Hello. Okay, thank you, students. Now you can leave the meeting.
Okay, ma'am.
Okay, thank you.
Thank you, ma'am.